that we have this virtual platform so that we can all get together and get to see some of the just top ALK experts. And tonight we have the pleasure of having Dr. Shaw here. It's just so exciting that we get to be able to do this. But I just wanted to kind of go over a couple things. In 2020, we did some amazing things, particularly through Lung Cancer Awareness Month. We raised $1.8 million together for ALK research. And so that's so exciting that as we work together, we can kind of contribute to some of these things and make sure that this disease is studied and that we find out more things for it. If you want to know more about that, you can check us out at ALK-positive.org. And if you have any questions, you can send all of your questions to info@ALKpositive.org. So I just wanted to let you know we're here. If you need a buddy, reach out to Alice Chu. She has the ALK Positive Buddies program going on. And there's so many exciting things that we have coming up in the next year. So make sure that you subscribe to our newsletter so that you can keep informed. And that's it. Thanks, guys.
All right, appreciate it. And Gina is our president of ALK Positive, which I appreciate everything that you've done in the past. Hold on, Mr. Steiner, can you please mute? Hold on. So anyways, I'm going to go ahead and turn it over to Colin. I just want to remind everyone during ALKtALK that we have weekly meetings also that please take advantage of. And Colin, I'm turning it over to you. Colin's our head of our medical chair. So Colin, you can introduce Dr. Shaw, please.
Terrific. So yes, Dr. Alice Shaw used to be at MGH. She's still a little bit at MGH. MGH is Massachusetts General Hospital. Alice Shaw is going to give a little bit of an introduction about how she got into the thoracic oncology space. What's really amazed me about Alice over the years is that she was a combination of an amazing clinician as well as being an amazing kind of leader in research. And they're two different things. You can be a great researcher or you can be a great clinician, but to be both of those things is really exceptional. And it's what really put Alice, I thought, as sort of truly a leader. And we had so many patients of Alice that would say, she was calling and texting with them at all hours of the days and weekends and nights. And that's just amazing. We know how busy she is. She's lecturing to other doctors. She's, leading research programs. She's seeing patients. And, people like Alice and Ross Camage and Christine Lovely, I swear they've got 48 hours in every day. I just don't know how they do it. So I'll hand it over to Alice. She'll do a little bit of a self-introduction. Please feel free to start typing your questions into the chat, and I will select the questions that seem most appropriate to read out to Dr. Shaw.
Thanks, Colin, for that very nice introduction. And it's great to be here tonight. I did a quick scroll through the frames and could see many of you, and it's so nice to see so many of you here tonight. So I'll give a little brief intro maybe on myself and a little perspective on the ALK field, but I don't want to say too much because I really want this to be a session for all of you to ask questions and have us be able to discuss what's on your minds. So I have been in Boston for a number of years, started my training here many years ago, and did my residency and fellowship training up here. And actually decided pretty early on in my oncology fellowship, so this is around 2000, so over 20 years ago, that I was interested in lung cancer. And I have to tell you that it was a very different field 21 years ago. What really drew me to the field at the time was that we knew so little about the underlying biology of lung cancers. And it was very clear that deficiency in our knowledge of lung cancer biology was the reason why we had so few therapies. It was just chemotherapy back in 2000. Literally, it was carboplatin, paclitaxel, maybe some second-line chemotherapy. And I remember being in clinic with my mentor at the time, who was Tom Lynch. And I remember thinking that this was horrible, that we had no options for patients. And so this really motivated me to want to, of course, be on the clinical side, but more importantly, to also want to do more research to try and understand some of the potential drivers of lung cancer. And so at that time, around 2001 now, I joined the lab of Tyler Jacks at MIT, and actually spent about 5 years studying KRAS-driven lung cancer. So KRAS is another oncogene, just like ALK is an oncogene. And KRAS, though, we've known about for many, many years, over 3 decades now, and yet no way until recently to target KRAS. So back when I was in Tyler's lab, I was trying to understand how KRAS was functioning as a driver in lung cancer, and learned a ton during that time about basic research and about how critical basic research is to basically leading to clinical discoveries. And around the time I was in lab, many of you may know that EGFR mutations were discovered in 2004. That actually was initially discovered by a few labs, including at Mass General. And so I was very plugged into that, and it was incredible to see that there were a subset of patients, about 10 or 15% in this country, who had a mutant oncogene that could confer just exquisite sensitivity to a targeted therapy pill. And so at the time, it wasn't exactly clear how this would all pan out, but the hope was that that was just the tip of the iceberg, and that there would be other oncogenic drivers like EGFR in lung cancer. And so that was really exciting at the time. It really showed us how, if you actually studied in the clinic what was going on and made observations, that could lead you back to the lab where some basic discoveries could be made, which then could go back again to the clinic to further advances. And so it was very clear with EGFR, the importance of molecular biology of lung cancers, but also this importance of doing what we often call bench to bedside and bedside to bench research. And that actually led me to then take on this translational position at Mass General in 2007. And I was really fortunate because when I first joined Mass General in 2007, a group in Japan had just published a paper in Nature, which many of you probably now have heard about or seen. And this was the first discovery or first report of ALK rearrangements in a small subset of Japanese patients. And we were, again, very excited about this because, again, back then, all we knew about was EGFR, and we knew there was a potential for there to be other oncogenes. And so this discovery really said, oh my gosh, there is another oncogene. So I worked very closely with John Iafredi at the time in 2007, and we developed the first diagnostic test, the FISH assay, and actually started identifying patients in our clinic who had ALK rearrangements. And again, there was just so much, I would say, serendipity here, because as we were developing that diagnostic test, we also did happen to have crizotinib, our first generation ALK inhibitor, in the clinic in phase 1 trials, already well into its dose escalation. And so we were very quickly able to identify patients and then start them on what we thought was going to be an ALK inhibitor. We didn't even know at the time how good it would work. And in 2007, actually right after Christmas, we enrolled our first ALK patient on the phase 1 study of crizotinib. And this patient had one of these amazing responses clinically. And that was the start, I think, of the ALK field. So we knew, think about it, back in 2007, end of 2007, that ALK was going to be a huge, important finding and advance for patients with lung cancer. And from there, we worked on crizotinib. And crizotinib, as some of you know, I mean, that really was a miracle drug for many patients. Some of you are on here and have been on crizotinib. But it became very apparent probably within about a year or two, so about 2009 timeframe, that patients were beginning to develop resistance. And so I worked very closely with a lot of colleagues at Mass General to now focus on this problem of drug resistance, which we know is not unique to crizotinib. It actually, unfortunately, plagues all of our targeted therapies and chemotherapies and even immunotherapy for that matter. And so really, we led a large research effort to identify mechanisms of resistance. And this helped us then in collaboration with a number of pharmaceutical companies to develop now second generation, more potent ALK inhibitors, such as ceritinib or Zycadia, Lectenit, Alecensa, and brigatinib, Alenbrig. And so this now led to clinical trials, phase 1 trials, again, where we were starting to see pretty amazing results with these new second generation inhibitors. And many, many patients of ours went from crizotinib to a second generation inhibitor and did amazingly well and got not just a few months, but sometimes a few years or more from each of these drugs. And then, of course, we had the problem of resistance developing to second generation inhibitors as well. And we worked very closely with Pfizer even before lorlatinib entered the clinic to understand the potency, the activity of lorlatinib in ALK patients, and the brain penetration of lorlatinib. And this really allowed us then to push the phase 1 study of lorlatinib through. And of course, many of you know that lorlatinib demonstrated really remarkable activity in patients who had been on prior ALK inhibitors, especially in the brain. And now we even have data on lorlatinib frontline, where it shows very impressive activity, especially in preventing brain metastases. So it has been an amazing ride, so to speak, from 2007 to now, right? That's about a 13, 14-year span where we have, 5 ALK inhibitors approved. We have multiple sequential therapies that we can use. And as some of you know, we have a number of combination therapies also in the clinic as well. So I think that gives me a lot of hope, just seeing how fast the pace has been in developing these ALK inhibitors, understanding what ALK-positive lung cancer is, and now developing new combinations. That there's a lot of research ongoing. We need more, of course, but there's still a lot of research that's going to continue to push the boundaries for our ALK-positive patients.
Fantastic introduction. So you mentioned Dr. John Iafrate, and so the timing on that's pretty unique. So Dr. Shaw just mentioned that she worked with Dr. Iafrate back in 2007. And ALK-positive has just given an award in the last few months to Dr. Iafrate for new research. So, that's just a wonderful circle. I love that story. You mentioned about lorlatinib being sort of the crown study, and undoubtedly will be approved as a first-line drug within the next few months. I think the big question on a lot of people's minds is, do you think it will become the universal first-line drug, or do you think it's going to be in specific situations it'll be used first-line? Dr. Kamich felt that he, at this stage, would not use it first-line. I just saw a video today with Dr. Sanjay in UK. He felt at this stage that he would hold it in reserve also. You might have a totally different perspective, so I think everyone here would be very interested to hear your perspective on that.
Yeah, I mean, I worked for a number of years on the CROWN study. We recently published the data. I've reviewed it with actually a few of you on the call, actually. The CROWN data is quite impressive. However, the limitation, as most of you know, is that the comparison was done versus crizotinib, because at the time we started the CROWN study in 2017, crizotinib was still the standard first-line therapy. And so all the comparisons we're trying to make between, for example, Alecensa and Mrna are through what we call cross-trial comparisons. Obviously, the ideal would actually be a head-to-head comparison of Mrna versus Alecensa as frontline therapy, but what we have is Mrna versus crizotinib. So we have to keep that in mind, that we'll be comparing the CROWN study versus other trials like the global ALK study, so 2 separate trials. But I think what's important to note, given that limitation, is that the benefit of lorlatinib over crizotinib was not just substantial, it was huge. And that magnitude of benefit was significantly greater than the magnitude of benefit that we saw with Alecensa over crizotinib or brigatinib over crizotinib. Just to give you a sense of what I mean, what we saw with lorlatinib is that the risk of progression or death was reduced by 72 up to 78% with lorlatinib compared to crizotinib. And in Lectenit and brigatinib studies, the risk of progression or death was reduced about 50%, 50, 55%. It's a significant improvement. And the other thing that was quite notable that is not trivial, and many of you, again, have talked to me about this, is that, for ALK-positive lung cancer, for some reason, the cancer has a propensity to spread to the brain. And we saw this very early on when we first started identifying patients with ALK-positive lung cancer, and why exactly ALK-positive lung cancers have that propensity, we don't understand, but it's true. And so treating and preventing brain metastases is incredibly important. And here I think lorlatinib has a clear advantage. It is the most potent and brain penetrant of the ALK inhibitors. And in fact, what we saw in the CROWN study was a very, very significant reduction in the what we call the cumulative incidence or the development of brain metastases. And in patients who had already metastases when they joined the CROWN trial, what we noticed, and again, I don't know how many of you might have seen this, what seems like a small detail, but it's important to us as clinicians, which is that over 70% of those patients who had brain metastases actually had a complete response in the brain. So lorlatinib completely eradicated the brain mets. However, with other drugs, alisenzimab, brigatinib, and ensartinib, the complete response rate is in the range of 25 to 35%, that type of range. So very different in terms of CNS potency. And I think the higher efficacy, we know it blocks more ALK mutations. We actually blocks all single ALK mutations. And then on top of that, it also prevents and treats brain metastases. I think that's the reason why that translated into such a phenomenal improvement over crizotinib. Again, I suspect it's better than Lectenit, but I can only say that based on cross-trial comparisons. But based on the data though, Colin, I would say that yes, I think lorlatinib will move into the first-line setting. The question around subsets of patients who may benefit is a good one. We did do, we had some pre-specified subsets in the CROWN study. And so we looked across these subsets. We didn't see any obvious ones, certain subsets of patients benefiting more than others. However, the molecular subsets are all still pending, meaning that could there be a greater benefit for a patient, for example, who has one type of ALK rearrangement versus a different type of ALK rearrangement? That sort of thing, we'll have that probably in the next 3 to 6 months this year. So probably at AACR, ASCO, those type of meetings, we'll have some of these subset analyses that will answer that question, Colin. That will be helpful to see. But I would say stepping back and just thinking about what the general sort of teaching is in oncology is we do want to use our best therapies upfront. We know that patients they do seem to derive the most benefit when we use our best therapies upfront. So I think that while people may not feel ready to make the change yet, people want to see some more of the subset analyses, see longer follow-up, see what the median progression-free survival will actually be. I think it's all headed the right way. And I think we will ultimately shift to lorlatinib because it is the most potent, it covers the most ALK mutations, and it protects the brain the best.
I don't know if I ever shared this with you, but we did an internal poll and we asked members of our support group that had been on both, not at the same time, but at different times, had experienced both alectinib and lorlatinib. And the question was worded, I don't have the exact wording in front of me, but it basically said, if all else was equal, if they worked equally well, and so on, which did you feel better on? And I was expecting that they were going to say alectinib. And we'd had so much feedback from people saying, my brain wasn't functioning right, and I was getting angry, and I was having mood disorders, and my cholesterol had gone through the roof and couldn't get brought down, and everyone was getting really, pretty uptight about the whole lorlatinib thing. And the current stance is, and this isn't quite exact, but it's something like about 45 people said that they felt better on lorlatinib, and 25 said that they felt better on alectinib. So you've actually got more people feeling better on lorlatinib than you do on alectinib. And I think it's a lot to do with energy levels.
Yeah, for sure.
And muscle pain and things like that. So
also, Colin, I do think it's really important to point out to everyone that, I think lorlatinib can be challenging for some patients. And I think oncologists really, and patients also really need to understand the importance of dose reduction. The 100 milligram dose that was chosen as the so-called standard dose was known when we chose it that it was going to be on the high side. And when we had the initial or the meeting for the investigators and the Pfizer team to discuss the dose to go forward with, there was a lot of debate. And just for what it's worth, I wanted 75 milligrams because in my experience, 100 milligrams was too challenging for the majority of patients. But I think the thought was, well, we want to make sure we can cover all of these mutations. And I should tell you guys as ALK patients that there was also this need to cover ROS1 mutations. And the ROS1 mutation we were trying to cover is basically impossible to cover, even at the 100 milligram dose. So they erred on the side of a higher dose. And in my practice, I do find that 100 milligrams is too high for most. And in fact, I typically start my patients at 75 milligrams. My patients do great at 75 milligrams and even 50 milligrams. I've gone down to with patients, even with G20, with our G1202R mutations and other ALK mutations, responding well at 50 or 75 milligrams. So the key here is that we can't be shy about dose reducing. Dose reducing absolutely helps mitigate the side effects and is still a very active drug. Basically, we saw the same activity at 75 milligrams and 100 milligrams, and yet we chose the 100 milligram dose, just so you know. So that's why I'm very comfortable starting at 75 milligrams and even going down if needed. It's an incredibly active drug, including in the brain.
So given what you just told us, and once it becomes first line, which as we said is probably inevitable, what's it going to take for people to just say, I want to start on 75 milligrams rather than 100? And not have to go through all that problem.
Yeah, I mean, I think, and to be honest, a patient who is newly diagnosed upfront, they absolutely should start at 75 milligrams. What it's going to take is just a patient to know that that's a possibility and an oncologist to feel comfortable doing it. Now, and I have every once in a while, I'll start at 75 and a patient will be doing so great, they will want to try 100. So there's no problem doing that. But sure. Most of my patients are pretty happy at 75 or may need to go down even to 50 milligrams. But I would say if I'm ever going to start a newly diagnosed patient, I would start at 75 for sure. So
given the prescription information is going to say 100, how's that going to get out to all the oncologists prescribing it?
Well, I mean, again, an oncologist doesn't have to prescribe 100 just because, they have to be familiar with the drug and know that the dose reductions are, in 25 milligram increments, and they can start a patient at any dose they want. Just as an example, for Tarceva, which is a first generation EGFR inhibitor, the standard dose was 150. But we typically started patients with the most sensitive exon 19 deletion at 100 milligrams all the time. So again, once I think oncologists are familiar with the drugs and understand that the drug is very active, even at 75 and 50 milligrams, I think that will become routine.
Okay, terrific. All right, I'm going to start reading through some of the questions here, Alice. So there's a question here from someone who says, what does it mean to have an ALK wild type progression? Is that equivalent to a loss of the ML4 ALK rearrangement? Does it mean that any potential ALK target is lost and we would have zero chance of responding to the ALK peptide vaccine or cellular therapy funded by ALK-positive?
That's an excellent question. It's really confusing, some of the terms that are used. So when if you're an oncologist or you're talking to one of the researchers, they talk about resistance in the setting of ALK wild type. Actually, everyone retains their ALK rearrangement. That's what we call a truncal driver. It's always there. But when they say ALK wild type, they're referring to the presence, or I should say absence, of having an ALK resistance mutation. And so it's not, the implication is that ALK itself is not driving the resistance, but there's something else besides ALK, an ALK independent mechanism of resistance that's active and accounting for relapse. So ALK wild type relapse refers to ALK independent mechanism of resistance. Now that may mean that for example, if you have Latinib and your cancer is beginning to progress and a biopsy is done and there's no evidence of an ALK mutation, your doctor may say that you have an ALK wild type. You still have the ALK rearrangement, you just don't have an ALK resistance mutation. And the chance of you going on then to respond just to switching to another ALK inhibitor is incredibly low, especially if you've just been on Latinib. However, your question around some of the other therapies targeting ALK, such as a vaccine, would still be relevant here because you still have the ALK rearrangement. You still are expressing the ALK fusion protein. So a vaccine targeting the ALK fusion should hopefully still be active.
Terrific. So next question here says, for patients allergic to the contrast used in X-ray computed tomographies, could this make it more difficult to monitor the disease properly? Are there other technical solutions for this type of situation?
Yeah, so that's a very good practical question. And this does come up because it's not uncommon to have allergies develop to IV contrast. And if you've been receiving treatment for a number of years and getting periodic CT scans, which you almost certainly would have with IV contrast, that sort of chronic exposure to IV contrast can also lead to you developing an allergy. And so this does come up in the clinic quite a bit. For my patients who have an allergy to IV contrast, and now we're only able to do scans without contrast, I do typically consider interdigitating a PET scan, because you do lose some sensitivity by taking out the IV contrast. As an example, for example, the liver, once you have no IV contrast there, it's much, much harder to detect lesions in the liver. And so for my patients who are getting regular CT scans, but they can't get them with contrast, probably every other or every third scan, I might include do it as a PET CT. And that just gives us some extra reassurance that we're not missing anything by just doing scans without contrast.
Terrific. And I'm going to throw in my own question here, since we're on the topic of PET scans. From your point of view, after initial diagnosis, when do you prescribe patients to get a PET scan?
So they've just been diagnosed and not even had a PET scan yet?
No, assuming they've been diagnosed, they've had their PET scan, but after that, when would you next prescribe a PET scan?
So certainly in the situation I mentioned, if there were someone who couldn't get contrast scans,
right.
But also, usually we don't monitor a patient routinely with PET scans. I really would do a PET scan if there was something on the CT scan that was of concern. And I felt like further characterization of that lesion or that area would be helpful by PET scan. Or it could be that a patient at some point, it's very clear on CAT scan that they've now progressed and we're about to switch therapies. Sometimes then I will also get a PET scan to do what we call restage, just get a sense in the whole body where there are now active sites of disease.
Terrific. Okay. So next question here, should a patient with brain metastases who is on a second or third generation TKI always stick to having an MRI every 3 months, even if the brain mets have been stable?
So it's a good question as well, because second generation ALK inhibitors have great CNS activity. And of course, Latinib has great CNS activity. And so we often do see these great responses in the brain. And sometimes you don't even see any evidence of brain metastases. For the most part, if a patient has had prior brain metastases and now they're being treated, I still do monitor the brain very closely, especially on second generation drugs, because patients absolutely will develop brain metastases on Alecensa, on Alunbrig. So I definitely will continue about every 3-4 month monitoring of the brain on those second generation drugs. And on Latinib, again, it's a very potent drug in the brain. However, every once in a while, we still will have a patient develop resistance in the brain on Lorlatinib. Fortunately, it seems that Lorlatinib controls the brain really, really well for most patients, but there is still that chance. And so I do frequently still monitor the brain. I would say I very rarely go to anything less than every 3 or 4 months in a patient who has had previous brain metastases, even if it seems like Lorlatinib has eradicated all of them. And the main reason for that is because, as some of you know, you can develop small brain metastases and have no symptoms, like not a single symptom. You can feel great. And they could be very, very easily treated with focused radiation, stereotactic radiosurgery, gamma knife. And it's much easier for us to treat a tiny asymptomatic CNS lesion as compared to a lesion that's now large and symptomatic. And so that's my reason for continuing very active surveillance in patients, even when they're on very brain penetrant ALK TKIs.
And what frequency would you do if the patient has not had a history of brain metastases?
Yeah, so that's a good question too. On Alecensa, I often, again, still survey the brain, probably every 6 to 9 months on Alecensa. And on Lorlatinib as well, I would survey the brain, again, probably similar frequency, 6 to 12 months, that type of time frame. I don't do it every scan in a patient who's never had brain metastases, but I do the surveillance. I have noticed that some oncologists will either not do them or maybe they just do them once a year. And I've had a number of patients where they developed early brain metastases before that year. And so now that's why I've dropped it, Colin, down a little bit to sort of more like 6 or 9 months.
Okay, terrific.
There's no magic answer here.
Yes, none of these have magic answers. So the next one definitely does not have a magic answer. With Lorlatinib being considered as a first-line treatment, what options are there for patients when resistance develops to Lorlatinib? And then I'm going to ask one more question to go on top of that question is, how do you then decide which of those options you would actually choose in different situations?
Yes, so it's an excellent question. And this is true for patients who are on lorlatinib first line or lorlatinib third line, right? So the whole question of mechanisms of resistance to lorlatinib is really, really important. When I was at Mass General, we did a ton of research around lorlatinib resistance. And I should note that all of the patients, every study we've ever published on lorlatinib resistance has been on patients who were on prior ALK inhibitors. And usually they were on sequential crizotinib, Alecensa, lorlatinib, or crizotinib, alectinib, brigatinib, lorlatinib. So they had received a number of ALK inhibitors in the past, and that definitely can shape and impact the resistance mechanisms that ultimately emerge. Now, in contrast, mechanisms of resistance to frontline lorlatinib are much less well understood. And even at my site, we've only recently, in CROWN, we had one patient on the CROWN study, and lorlatinib's not even approved first line yet. So we really don't have very much experience with patients on first line lorlatinib. Plus, as I just mentioned, the CROWN data were incredibly positive. Patients have done incredibly well on first line lorlatinib. We don't have a lot of progressors on first line lorlatinib. And so we don't understand mechanisms of resistance yet to first line lorlatinib. But based on all the laboratory research that we did, we would predict that in contrast to patients who've been on sequential ALK inhibitors, where we know that ALK resistance mutations can accumulate in that setting, when patients receive first-line lorlatinib, we would predict, and the laboratory studies validate this, that we may not see any ALK resistance mutations emerge because lorlatinib is so potent across all of the single ALK resistance mutations. But we know that resistance likely will develop at some point. So then what are those mechanisms of resistance? We don't know from the clinic yet, but I would say that our data probably suggests that it will be ALK-independent mechanisms, meaning that other targets, other kinases, other growth signaling pathways that get turned on aberrantly to allow the cancer to now evade lorlatinib. Lorlatinib is still able to shut down ALK, but these other pathways get turned on and now can basically take the place of ALK. One of the ones that I think will become important in the setting of frontline lorlatinib resistance is MET. We have heard of that anecdotally, and we have certainly seen that in patients who've been on sequential ALK inhibitors, and then to lorlatinib, they now have high-level MET amplification driving resistance. So MET is an important one, and that's the basis for a number of combination studies that I'm sure many of you have heard about, where we're combining ALK and MET inhibitors.
Terrific.
And then just to that question, Colin, about how would you pick sort of next options, I definitely always recommend, if a patient's developing resistance to any ALK inhibitor, be it first line, be it third line, or whatever, that there's some effort made to do a rebiopsy. That could be a simple blood rebiopsy, a Guardant test, for example, but it could also be a tumor rebiopsy, because this is how we learn about what could be driving resistance. And sometimes this can point you in the direction of a particular combination or a trial. And that's how I would make a decision about what to do after lorlatinib.
Thank you. Thank you. So next question, while lorlatinib plus MEK, and I'll spell it out so it doesn't get confused with MET, MEK, and SHP2 inhibitors would probably be too toxic for a combination. Is there any rationale for an intermittent use of MEK and SHP2 in addition to lorlatinib?
Yeah, so I think that question is referring to the trial that we have at Mass General, which is combining, it's two drug combinations, lorlatinib with a MEK inhibitor, binimetinib, and then there's a separate combination of lorlatinib with a SHP2 inhibitor, TNO155. I think even developing these two drug combinations is going to be probably a little challenging in terms of toxicity. So I'm going to be curious to see how, as we go up on the doses, especially of the lorlatinib MEK combination, how patients are doing, because even a two drug combination may require some intermittent scheduling. The three drug combination is interesting, and certainly there's preclinical data around combining SHP2 and MEK inhibitors, especially in KRAS-driven cancers, but potentially there's a lot of toxicity hitting that pathway so hard. And so would we explore that? We could, but I would want to get through the two drug combinations first before thinking about a three drug combination.
Terrific. Is it advisable to combine Lectenit with another treatment, for example, radiotherapy or immunotherapy, in order to get a better response?
So, yeah, that's a complicated question, but I would first say that right off that we would not combine Lectenit with an immunotherapy. In fact, we wouldn't combine any ALK-TKI with an immunotherapy. I'm sure there could be some exceptions here and there, but in general, we wouldn't, because targeted therapies just do not mix with immunotherapies. It's interesting because Lectenib is generally so well tolerated. Immunotherapies by themselves are so well tolerated. But when you start mixing the targeted therapies with immunotherapies, we see a lot more toxicity. You basically see this enhancement of the TKI toxicity. So when we combine Lectenib with immunotherapy in the clinical trial, it didn't appear too bad in terms of the numbers, but anecdotally, I had a number of patients on Lectenib and immunotherapy, and you had many more issues with skin toxicity like rash, and also hepatitis or transaminitis, so liver irritation as well, and also pneumonitis. So I've had patients struggling with all of those pretty serious toxicities when we tried to combine Lectenib with immunotherapy. So I would say no, we would not do that. And there's also no preclinical or clinical evidence that adding immunotherapy would help the Lectenib at all. And because of the toxicity, if anything, it's going to hurt the Lectenib, because it may force patients then to have a dose hold or a dose reduction because of that toxicity. In terms of Lectenib and radiation therapy, again, we don't have great data combining TKIs with radiation therapy, but some of you know that we will use them sometimes, not so much in combination, but we'll use them together in kind of a multimodality approach, where we may have a patient who went on Lectenib, had a fabulous response, but they have one little residual site of disease in the lung. And in that case, you know, our thought is that perhaps there's some dormant cancer cells in that site of residual disease that could later develop full-blown resistance. And so we will oftentimes think about trying to eradicate residual disease using radiation therapy. And so that's what I mean by combining. I don't necessarily mean doing Lectenib radiation at the same time, but we often use them in conjunction with each other. And the radiation, the hope is that we can eliminate any residual disease that could be sort of the precursor for resistance down the road.
So Dr. Camage had mentioned that his idea on local consolidative therapy was, or basically the way, his method for determining the point of maximum effect of a second generation drug such as Lectenib is when he's had one scan and then the next scan shows no further improvement, no further reduction in size. And at that stage, he feels that that might be a pretty good time to be doing radiation local consolidative therapy. What's your thoughts on that? And would you consider doing that routinely? Do you think that might become a standard of care?
Yeah, I mean, we do it. I mean, I have to say, we do think about it essentially for every patient, and not just for frontline Lectenib patients, but if I have a patient who now went on Latinib after Lectenib and they're having a great response, but there's some residual disease, we're always thinking about that. Can we use radiation to eradicate that? You know, the question of timing is a good one. We don't know the optimal time to do the radiation. We have a protocol at Mass General where we are doing consolidative radiation therapy in patients who are on a EGFR, ALK, or ROS1 TKI, and we're actually offering that treatment if they're within 6 months of starting the therapy. And so I think what Dr. Camage mentioned is reasonable. You know, we want to do it when we think you're at maximal response. And so that can happen for some patients after the first 8 weeks. Sometimes it's after the first 16 weeks, or sometimes a little later. So it's hard. You can't set an exact time frame for every person. So that's why we follow scans. But typically we'll do it within the first 6 months.
Terrific. And we get a lot of, especially the newer members, and they, you know, read and get told by friends that, you know, food, diet, lifestyle, sugar consumption, you know, I'm sure you've heard all these things, you know, over and over and over and over again, that these things can have an effect on the actual cancer, not on how you feel or, you know, just how you're dealing with things, but on the actual cancer. So do you have any thoughts that any of that could have any impact on the actual cancer process itself?
I guess the bottom line is we don't know for sure, but why not try and be as healthy as possible, right? Because yes, could there be an impact on the cancer? Possibly, if you're eating super healthy and you're exercising and you're active. But I do actually think what's equally important is how you feel, right? And all of us, I think, would agree that quality of life is incredibly important. So if eating well and exercising is going to allow you to feel well, and also I would say doing that also allows us often to tolerate the drugs that we're on better. Like, why not do it? So I do talk to most of my patients about diet. For patients who are on Alecensa or Lorbrena, we know weight gain is a big issue, and weight gain can certainly make you not feel great. And so we often talk about strategies to modify the diet. Limiting your carbohydrate intake, for example, is a big help for Alecensa and Latinib. The other thing is exercise. I mean, there are studies that suggest that activity and exercise do actually have an impact on your cancer. But again, I think what's more important to me is it allows patients to feel better. And some of the side effects, like the weight gain I mentioned, but also the edema that both Alecensa and Lorbrena cause, those oftentimes are improved in patients who are exercising regularly. So there are many, many benefits to exercise. Yes, possibly for the cancer, but more importantly for your overall health and your quality of life.
Great. I'm sure you were expecting that you'd probably get a couple of questions about COVID vaccine and whether it would interact with TKIs. So we've got a couple of questions about that. So I'll let you speak to that issue.
Yes, so we are very, very thankful that the COVID vaccines are rolling out, maybe a little slower than we would like, but they are rolling out. And certainly I think patients with cancer should receive a COVID vaccine. Many of you probably know that these are what's called mRNA vaccines. These are not the virus itself. It's not an inactivated virus. It's an mRNA that's encoding some portions of the spike protein of the coronavirus. And so far from the trials that we have, all the data that we have, these vaccines look quite safe. And I did listen carefully to the ASCO recommendations, and ESMO has recommendations as well. And I think everyone agrees that while cancer patients were not included in the trials of these COVID vaccines, I don't think there's a reason to think that cancer patients would have, more problems, especially patients on targeted therapies, for example. There's no reason to think that they're going to have more problems with the vaccine, the COVID vaccine. And in fact, the net benefit is that you're going to be protected. 94, 95% efficacy with these vaccines. So you're going to be protected from having COVID and having a severe COVID complication. So I think we always think about risk benefit. Yes, we don't have all the information we want to have on COVID vaccines and cancer patients, but we do know they're very, very effective and that they will be able to prevent COVID infection. And so, yes, I would say for patients, ALK-positive patients, once the vaccines become available, I would think that all of our patients, with rare exceptions, all of our patients should receive it.
Terrific. And you had mentioned at the summit, and most of us, or many of us, are aware of it from other sources as well, that there's a so-called fourth generation TKI coming out I shouldn't say coming out, but is in trial from Turning Point Therapeutics. Do you have any knowledge about where that's at, or what you think of it, or just anything you would be willing to say about it?
Yeah, I mean, there's some data in the public domain. Some of you may have seen some of the data presented at AACR. I think it's TPX0131. And actually, we had worked with Jean at the time and shared with her these compound mutations that we were seeing in the clinic to allow her to focus in on some of the sort of common ones. I think that the data so far that's out in the public domain looks promising. TP Therapeutics obviously is making these targeted therapies. They know how to make drugs. But for me, the complicating thing for our ALK patients is that there's not just one compound mutation that develops in all patients. What we've seen actually is a large, diverse array of compound mutations, sometimes 2, sometimes 3 compound mutations. In fact, a year ago, when I was looking at the data, and I looked at it again recently, but I didn't see a single patient at that time with an overlapping compound mutation. Now that I've looked at the data more, we have some with overlapping mutations, but in general, because there's so many single ALK mutations, you can imagine all the different combinations that a cancer could develop. And so to imagine that one drug could overcome all of those compound mutations, I think is going to be hard to do. And so then you may need to have more than one fourth generation drug to be able to capture the majority of these ALK mutations. So Colin, I don't know much more than probably you guys know about it, but I think it's not going to be as simple as you know,
Right,
had a second
right.
generation drug to overcome crizotinib. We had Mrna to overcome some of the mutations, but we're not going to have just a single fourth generation drug that can really cover all of the compound ALK mutations.
So I'm going to read out this next question literally, and then I'm going to sort of add my little extra bit at the end to narrow it down a little bit. But the question is, if I can find it here, what does Dr. Shaw think is the most promising potential treatment for ALK? So let's sort of look past TKIs and look at other things such as combinations of ALK TKIs with other drugs such as what Dr. Bavona and Dr. Lovely are researching, or immunotherapy such as what Dr. Gaynor and Nemanoff and White have been researching, or any other fields. Which do you think is likely to end up being the one that ends up having the best chance of making a breakthrough?
Yeah, so I mean, I think ALK inhibitors, ALK TKIs are the most important thing for an ALK patient. So let's just put that out there. There's no question that Alecensa and Mrna, these are sort of the anchor molecules for any patient who has ALK. But because resistance develops, we do know that those are not enough. And so I do think it's a very good question, like what is the best? We don't know yet, but it almost certainly is going to be some type of combination. And I would say it's going to be likely what we often call cross-platform combinations. So it's not going to be probably as simple as just combining an ALK and a MET inhibitor and thinking, okay, that's going to be transformative. That could be a combination that we do take forward, but that's even not going to be enough. I'm talking about combinations where we utilize an ALK inhibitor to kind of maximally reduce, and then we come in kind of in what we call an orthogonal approach. Maybe we come in with a type of immune therapy or vaccine, or maybe we come in with an ADC, or maybe we come in with chemotherapy even, to now try and eradicate sort of these what we call drug tolerant persister cells that are somehow able to survive even though they're being exposed at full efficacious doses to the TKI. And so I am excited about the combinations, but I do think we have to think beyond targeted therapies, and we have to be willing to use different therapies that aren't typically used necessarily in ALK patients.
Great. There's a question from a member in Europe, which means I'm pretty sure it's like crazy hour in the morning in Europe right now. So, and we've got some members. I saw the name of one that's in the UK. I've got another one here in Germany. So yeah, you've got people staying up at all hours to see you, Alice. And I think currently we've got 234 people on board. So pretty impressive. So the question is, do you know of patients who have had a full response on Lectenit and progressed later in the body? Does reducing the dose of Lectenit have an impact on individual PFS?
So the patient, maybe Colin, if you
had a full response, meaning a complete response, and then they progressed later in the body. And the question is, do you know of some patients that have had that? And I'm sure the answer, of course, is yes.
Yes, unfortunately, we do have that. You can have these amazing responses to a CR or be in that NED state that everyone talks about and still relapse. And, I can see that is concerning, because we were just talking about eradicating residual disease, but if you can't even see it, then how do you eradicate it, right? So that brings up that point. But I'm not sure what the point was about lowering the dose, Colin.
So does reducing the dose of Lectenit impact the progression-free survival time?
Yeah, so I would say Lectenit, yeah, I'm not sure if it's related to this question about CR, but I would say in general, the dose of Lectenit at 600 milligrams twice a day, which is standard, is a great dose. But it is kind of like, in my opinion, it's efficacious, but like right at that efficacious boundary. And so if you start to drop it too much, especially down to 300 milligrams twice a day, that is too low. And in fact, I do see more CNS relapses, for example, at the lower doses. I don't know if there's published data on PFS in relation to the lower doses of Lectenit, but my anecdotal experience has been that 600 milligrams is really the best dose. And some of you know that we did a lot of work early on to show that even going to 900 milligrams of Lectenit was very helpful in the setting of CNS relapse.
And just so that you're aware, and you may be aware or maybe not, but we're actually going to be rolling out a longitudinal survey very shortly, and it should be able to answer questions such as reduced dose impact on PFS. So we should have some pretty good data on that for you soon.
It's a good point though, because quite a few patients have dose reduced on Lectenit to 450 or 300 a day, twice a day, and often the question is, should I bring the dose back up? And that comes up a lot. It really depends on what the toxicity was, which led to that dose reduction. I would say a common one is LFT or liver function test elevation on Lectenit that then leads to some dose reductions. Again, there's no real published data on this, so this would really have to be done pretty cautiously with your oncologist, but I have in patients who now have stability on their lower dose of Lectenit for some time, 3-6 months of stability now on their lower dose, we will sometimes consider dose escalating again. Again, a lot of the rationale for that really is to try and treat or prevent brain metastases.
Perfect. So what are your thoughts on using chemotherapy agents such as ALKtA to treat oligoprogression in between or in conjunction with an ALK inhibitor?
Yeah, so for oligoprogression, I have to say our probably preferred method would be radiation if there's a lesion or lesions that can be easily radiated. But sometimes there's not. And sometimes then if that's the case, we could consider adding ALKtA. I would say typically though, if someone's on Alecensa or Allenbriggs, so they're on Lectenib or brigatinib or ceritinib even, and they have oligoprogression, if we can't use radiation, then usually we'll think about just going to lorlatinib because it is more potent and it's actually been demonstrated to have activity in that setting. And then on lorlatinib, if someone's having oligoprogression and we can't use radiation, then yes, we will sometimes add chemotherapy such as pemetrexed or carboplatin pemetrexed to the lorlatinib.
So having been on the academic side of ALK research and now being in private industry and the pharma side, what are the main findings or insights that you could give about kind of where things work from the pharma side? Any insight on what can be done to accelerate the journey toward making ALK a chronic disease or finding a cure? Is there a cure in sight in 5 or 10 years?
Well, many questions in that one. In my introduction, I didn't really speak to my transition to Novartis, but after being at MGH for almost 20 years, I did transition to Novartis last year, so about a year ago, to head up the early clinical drug development in oncology. So I'm in the R&D side of Novartis. It's called NIBR, based in Cambridge, right across the river from Mass General, which is why I still can go over and see some of my patients at Mass General. I've been there a year now, and it has been a huge learning experience and a very positive one because, while I was in academics, I worked with a lot of pharma and biotechs, and certainly we developed a lot of these ALK inhibitors together. But to be honest, from an academic standpoint, you don't actually know how drugs are made, where they come from at all. And so now being on the pharma side and being there a year, it's been incredibly eye-opening and fun to think again about science, and also to think very broadly, not just focused on lung cancer or just ALK-positive cancer, but to think very broadly, because honestly, you can take a lot of learnings from other cancer types and hopefully apply them to ALK. And so I have learned a ton about drug development. I've learned the importance of basic science and doing good basic science to really inform clinical drug development. And I have learned also the challenges of drug development, more so on the pharma side than on the academic side. It's really hard to make a drug, to make a good drug. And I would say combinations, which is what we're all aspiring to, and we need those combinations, are even harder. And I think here combinations are going to be informed. We need the best research to inform the combinations, right? We don't want to be guessing on combinations. These combinations are hard to develop. There are toxicities that patients have to sort of tolerate. We have to pick the best ones. And so really doing the research and having great preclinical models that are hopefully predictive of efficacy, and also doing some preclinical models to predict safety or toxicity is also really, really, really important. But I do think that what I've seen this last year is that there is an incredible amount of knowledge and innovation and potential now. And again, I see it more clearly now that I'm on the pharma side than when I was on the academic side. And I'm so excited to be able to be part of that. And hopefully, again, my goal in going to industry was to learn about drug development and also to learn about how to apply all of this to certain segments of lung cancer, like ALK-positive lung cancer and ROS1 positive lung cancer. And I think I'm going to be able to do that.
So Dr. Kamij spoke at length about tumor markers, and I'm sure you're aware of his philosophy on that. What are your thoughts on that?
Yes, so I know he follows tumor markers. I know some oncologists do. The reason they aren't widely followed is because we know that tumor markers can be falsely elevated in the setting of various non-neoplastic conditions. And so they can lead then to a lot of anxiety, a lot of extra tests, and so they can be very challenging. However, for some patients, they can track the cancer very well. And that could give you then sort of an earlier sense of when relapse would be happening. You know, we actually in the phase 1 LDK or Ceritinib study actually did track tumor markers in that group of ALK-positive patients on the phase 1 study of Ceritinib. And I had looked carefully through that data. And actually, it's exactly what I just summarized, that actually for a minority of patients, probably about 20-25% of the patients, we saw that the tumor markers tracked pretty well. So when there was relapse, you could see it go up, and then it went down with response. And then when there was relapse again, it went up. Perfect. But in the majority of patients, either the tumor marker was uninformative, like nothing was really happening, or it was not tracking at all. And so that gave me some pause that, you know, if the majority of patients, the tumor markers aren't tracking, then that could definitely give us some false hope about, or false concerns. And so I do see that if patients, if I'm seeing patients and they're already tracking their tumor markers and we have a sense that they're tracking pretty reliably, of course, we're going to continue to follow those tumor markers. But I don't routinely start all of my patients on this, checking their tumor markers.
So I know that you said you only had about an hour available, so it has been an hour.
We had 8:10. How's that, Colin?
Perfect. We'll go as long as you can go.
When you start hearing my kids or my puppy howling in the background, you'll know.
I'll tell you what Dr. Kamij did. Dr. Kamij had his daughter came in, waved in front of the audience, got told that there was 200 people watching, and then she bolted out of there.
Thank you, Dr. Shaw.
How can we encourage pharma companies to fund more combination trials with ALK inhibitors?
Yeah, that's a great question. I would love to, it's hard for me to say because actually Novartis, of course, made Ceritinib or Zycadia, which is not one of the major ALK inhibitors at this point because of its toxicities. The main pharma, of course, to approach would be Roche Genentech for Alecensa and Pfizer for Lorbrena. And exactly how you do that, Colin, I don't know yet. I'm still learning the ropes myself. But I would like to see that done because, so when I was still at MGH, we developed this combination study with lorlatinib. It's 3 arms actually, lorlatinib with crizotinib for MET inhibition, lorlatinib with MEK, and lorlatinib with SHP2. And they were very, very nice in basically supporting this study of ours. And the study is going great. However, you can imagine that if this was a sponsored trial, meaning Pfizer was doing this trial, and they now had it open even at 5 sites, not just 1 site, you can imagine it would go that much faster. And of course, it would give more patients access, right? Patients, and especially in the COVID era now it's just been so hard to have patients travel for a trial like that. And so I think somehow working through, all of the pharma do have patient advocacy groups within them. And so I think reaching out to those folks would be very important, and just connecting and explaining the need. I have had the sense now being in pharma and I had the sense as well being in academia that people think that ALK patients are doing great and they have 5 ALK inhibitors. They have some combinations and trials. Like, how do you get in that space? And I feel, and I worry about that because I think we all know that of course we need more therapies and better therapies. And so sharing that experience, everyone sharing their experience, especially if you work with these other patient engagement groups at pharma, I think would be helpful, just as education that ALK patients are still in need.
Right. Thank you for saying that. Here's a really interesting question. Would you recommend switching to lorlatinib if doing well on a Lectenit?
Yeah, so this came up a ton as well when global ALKs first came out as well, that I had many patients on crizotinib and they wanted to know if they should all switch to alectinib. So we've dealt with this in the past. Every time there's like a new thing, there's always that question, should I switch? I kind of handle it on a case-by-case basis. I would not say that routinely we should be switching everyone. Once it's approved, switch them to first line. I don't think that's the case. As an example, I have a number of patients who've been on crizotinib 5, 10 years even, and I didn't switch them at that time to alectinib, and they're still doing well. So for patients who have been on long-term alectinib doing well, I almost certainly would not switch them. I think the harder question is if they just started on alectinib, should they consider a switch to lorlatinib? And again, this is where I would take it case by case. I think one particular subset of patients where I might consider that is a patient who has brain metastases or leptomeningeal disease at baseline, where CNS control is critical. And as I said, I think the 600 dose of alectinib has a little bit of weakness in terms of CNS control. And so that subset of patients, Colin, I might consider making a switch.
Thank you. There's a question here, and I should know what the Alchemist trial is, and I can't recall. But the question is, I'm about to end the Alchemist trial. Are there any findings or results so far?
Yeah, so the Alchemist trial refers to an NCI-sponsored trial that was looking at the use of TKIs as adjuvant therapy after resection. So EGFR patients who were resected went on to an EGFR TKI versus placebo, and ALK patients who were resected went on to crizotinib or placebo after they had their chemotherapy. And no, we did not have results other than to say that it's been hard, I heard, to find ALK patients, resected ALK patients. And then on top of that, to use crizotinib as the adjuvant therapy. If I have a patient in clinic who's been resected, they're found to be ALK positive, I absolutely wouldn't choose crizotinib for all the reasons that you guys know. And if I were to choose an ALK inhibitor, I almost certainly would choose Alecensa.
Great. So I'm just scrolling through the questions here to try to find ones that are good ones to ask. Here's a real quick answer. Just give a real quick question. Just give a real quick answer so that you can move on to another question. Have you seen links to cardiac issues, specifically CHF, and I do not know what CHF is, from alectinib?
So CHF is congestive heart failure,
okay.
Or cardiomyopathy sometimes is what it's called. We haven't seen any drop in ejection fraction, which we can tell you about the possibility of CHF with Alecensa. Now, that's based on clinical trials and my own experience. Could there be a case here or there reported somewhere? Possibly, but I would say no, we don't generally think about ALK inhibitors causing cardiomyopathy. There really isn't expression of ALK in cardiomyocytes, and so it's not clear what the sort of basis for that would be. Now, for lorlatinib, I would say here was a little more complicated because lorlatinib causes a lot of fluid retention. And that extra fluid in the body can place some strain on the heart. And so if someone has underlying heart disease, I have seen it worsen that underlying heart disease. Mm-hmm. But in general, I don't think we believe that ALK inhibitors should cause CHF.
So just a quick run on from that question then. So when lorlatinib becomes approved as a first line, are there situations if a patient did have underlying heart conditions that you would maybe consider not putting them on lorlatinib and putting them on alectinib instead?
Possibly, yes. I would say yes. I would also say for patients who have a known and significant mood disorder at baseline, I also might not consider frontline lorlatinib because of its known side effect of causing mood lability. So there are a few cases for sure, Colin, where I would think very carefully about using Mrna first line.
Okay. And I'm going to and you can pass on this question if you want to. I have no problem with you passing on it. We're going to be having Dr. Awad speak with us in about a month's time.
Oh, good.
Next week's time. But the question is, where are we at in relationship to the vaccine for ALK-positive?
Yeah, so
you can pass on that.
Yeah, I'll probably pass it to him only because he knows the best. But when I last asked him, and I talk to him regularly, so when I last asked him, I believe that he was thinking 6 to 9 months from now. But we'll let him clarify that when he's on.
Yep.
I see a question, Colin, about potentially if you're doing well on Lectenit, going up to 900. Can I answer that one?
Sure.
Yeah, I think if someone's doing great at 600 of Lectenit and they've been doing fine for a while, I don't feel the need to bump up to 900. 900 does come with more toxicity. It comes with more fatigue, a little more muscle aching, sometimes a headache. I really only bump up to 900 when a patient has some evidence that there's some change in the brain. So a new brain metastasis or slight worsening of existing brain metastases.
So I know you have to go very, very shortly. So I'm going to ask this question just because it lets us kind of end on a high note of hope and moving forwards. And I'm going to throw in my answer before you throw in your answer. Okay. The question is, has the life expectancy increased with ALK-positive? What is the average after diagnosis? And I gave a comment the other day to someone's post, and just my comment, which is usually buried, it's not like a post, it's just buried. And, it was someone that was a new member and they were feeling, kind of a bit down and out about the whole thing. And my answer was, if I had been diagnosed in 2006, I would have been told I've maybe got a year left. If I'd been diagnosed when I was in 2016, the stats pointed to maybe 2 or 3 years. And now you're looking at stats that say probably about 7 years, and they're retrospective stats, which means that, it's probably even more than that. So my answer was, it's reasonable to expect, in another 4 years, it'll probably be, 10 years or whatever. And for another 4 years after that, it'll be even more. So that was my answer, but I'm going to let you answer so that we can finish on a high note.
Yes. So, actually, I do get this question a lot as well. And some of you may know that way back, like 2008 and 2009, when we were first identifying ALK patients, I actually looked at this, the question of how do ALK patients do? And this is really before crizotinib was even available, right? So before the ALK targeted therapies became available. And I can say that when we looked back at patients, this is what we call retrospective analysis. We saw that ALK patients who never received targeted therapies did not do well at all. They did the same as other lung cancer patients. And I think many of you appreciate, in general, lung cancer patients don't do well because of the absence of effective therapies, especially back then. And what we've seen is exactly what Colin has said. Every year, it seems like there are new studies showing that, in fact, survival is getting longer and longer. And so, in fact, Colin, when people ask me now, I don't even say a number because we've seen that it increases every single year. And so you said 7 years, I've seen 8 years. We've all seen it extending. And my hope is we won't have a real number because that's a sign that we're just getting longer and longer. It's unstable because patients keep doing well. And that's exactly what we want.
And I had heard initially that ALK is actually one of the more aggressive forms of lung cancer.
Right, it is.
If anything, the average is actually probably less if you didn't have therapies for it. Would you agree with that?
I would agree. And I would also say what supports that as well is that we tend to not identify ALK-positive disease in patients who have non-metastatic disease. I think it's a sign that the cancer tends to be metastatic very early. It's aggressive and it's metastatic early. And so, yes, so these numbers that keep increasing and getting better and better, I think is it's incredible promise. And it really just shows you where we're going.
Do you have time for another 300 questions?
Probably don't. But I do want to say that I'm happy, if there are any certainly urgent questions or anything, please, many of you have my email, feel free to reach out. Many of you know that I work very closely with Jessica Lynn, who is wonderful, and Jess and I are very close. We stay in touch. We talk about all of our patients who we now share. And so please feel free to reach out to me or to Jess at any time.
Terrific. Well, I personally can't thank you enough for coming on, and I'm sure everyone here wishes we could spend more time with you, and maybe we can spend some time again with you at some point in the not too distant future.
I would love that. That'd be great.
So I'll let Mark and Gina just sort of wrap up super briefly and let you go.
Go ahead, Gina.
Yeah, do you guys all want to unmute yourself and just tell Dr. Shaw thank you? Thank you, Dr. Shaw.
Thank you, Dr. Shaw.
Thank you.
Thank you so much.
Thank you.
Thank you.
Thank you.
Thank you.
Dr. Shaw.
Thank you very much.
Thank you so much.
Thank you.
Been great.
Thank you.
Thank you. Hey, want to say hi?
Thank you,
Hey,
Dr.
where's my dad?
Shaw.
Hey,
Love your
say hi.
babies,
Hurry up.
Becky. Love your babies.
Come on, say hi. Okay.
Thank you.
Thanks,
Thank
Alan.
you very much.
Very, very welcome, everyone.
These people.
Thank
Thank you, Dr.
so
Shah.
much.
And putting smiles on all our faces.
Thank you.
Thank you so much, Dr. Shaw.
Very welcome, Gina.